Okay, let's unpack this. If you ask a molecular biologist, what's the one process in a cell that's absolutely essential but also t- completely misunderstood?
They'd probably say water transport.
Exactly. Water transport, we all sort of assume water is small, the cell membrane is lipid, so it just, leaks through. A bit of simple diffusion.
And that's the perfect place to start this deep dive, because that assumption, it hides a huge functional problem.
Right.
I mean, water can diffuse through the membrane, sure, but it's incredibly slow. You've got this polar molecule trying to get through all these non-polar lipid tails.
Not an easy path.
Not at all. And then you look at tissues like the kidney or your salivary glands, and they have these water permeability rates that are, well, they're astronomical, way beyond what slow diffusion could ever explain.
So we're talking about massive fluid movements, right? Things that need to happen in milliseconds. This isn't just leakage.
No, this is a controlled high-speed plumbing system.
A plumbing system, I like that.
It is, and the machinery that solves this problem is the whole focus for us today. Aquaporins. These are the dedicated water highways, and their discovery really changed everything.
So our mission today is to quickly touch on the basics, the forces that push and pull water around, and then jump right into that revolutionary Nobel-winning discovery of aquaporins.
And we'll get into the genius of their structure, how they work, and then connect all of that directly to, real-world stuff like IV bags and even how plants survive.
Perfect. So let's lay down the basics really fast. We assume most of you know about diffusion, right?
Yeah, solutes moving from high concentration to low. But osmosis is the key here. It's the movement of water, the solvent, across a membrane.
And in a cell, that membrane is picky. It won't let big things like salt or sugar just waltz through.
Exactly. So the solutes stay put, and only the water can move. And water flows to where there's less water, which is always the side with more stuff dissolved in it.
It's trying to dilute the more concentrated side.
That's it. And that flow, that force, is called osmotic pressure.
Okay, so that pressure is what makes things happen to the cell. Let's talk about tonicity.
Right. If you put a cell in a solution that has the same solute concentration as inside the cell, we call that an isotonic solution. Think 0.9% saline for a red blood cell.
Iso means same. So water moves in and out at the same rate.
Yep. No net change. The cell is happy. It's stable.
But what if you drop it in, say, distilled water? That's a hypertonic environment.
Now you have a huge difference. There's way more water outside the cell than inside, so water just floods in.
And for an animal cell, which is basically just a little bag, that's bad news.
Very bad news. It swells up and it bursts. That's called lysis. It's why you can't just give someone an IV of pure water.
It would literally explode their blood cells. Okay, so conversely, what if we go the other way? A really salty solution, a hypertonic environment.
Now the situation is reversed. There's more water inside the cell than in the super salty solution outside.
So water rushes out.
It rushes out, and the cell shrivels up. It crenates.
And for plant cells, this is a bit different, right? Because they have that rigid cell wall.
That cell wall is a game changer. In that hypotonic solution where the animal cell bursts,
Yeah.
the plant cell just gets really firm and rigid. It fills up with water, pushing against the wall. That's called turgor pressure, and it's actually the ideal state for a plant. It's what keeps them standing up.
And in the hypertonic salty solution,
The cell wall stays, but the membrane inside pulls away as the water leaves. That's plasmolysis. You can see it under a microscope. The plant wilts.
so this all brings us back to the main problem. The direction of water flow is simple osmosis, but the speed, that's where things get interesting.
Precisely. The rates we see in tissues like the kidney are just orders of magnitude faster than simple diffusion could ever account for. And this is where the discovery of aquaporins comes in. It's a great story.
This was Peter Agre in the early 90s, right? He wasn't even looking for a water channel.
Not at all. He was at Johns Hopkins trying to isolate the rhesus blood antigen, the Rh factor, and his team kept finding this contaminant protein, a 28 kilodalton protein they called CHIP28.
And it kept showing up in places with high water flow.
Red blood cells, kidney tubules. It was a huge clue. But the real aha moment was the definitive experiment.
Right, the frog eggs.
The frog eggs. Xenopus laevis oocytes, their membranes are naturally, almost perfectly waterproof.
A perfect test subject.
The perfect control. So they took the mRNA for this CHIP28 protein and injected it into some of the eggs. Then they put all the eggs, the normal ones and the injected ones, into a hypotonic solution.
So pure water.
Essentially. The control eggs just sat there perfectly fine. The ones injected with the CHI-28 mRNA, they inflated like balloons and exploded almost instantly.
Wow, so that was it. That was the proof.
That was the smoking gun. It proved that CHI-P28, which he renamed Aquaporin 1, was a dedicated high-speed water channel. It was a massive breakthrough, earned him a share of the Nobel in 2003.
And the speed you mentioned
Yeah.
is just staggering.
It is. Each one of these channels can conduct about 1 billion. That's what B water molecules per second.
1 billion per second. That's not a leaky faucet, that's a fire hose.
Yeah.
But how does it do that so specifically? I mean, a water molecule is tiny, but so is a proton. If protons get through, that would wreck the cell's whole electrical system.
And that is the true genius of the structure. It's a masterpiece of biophysics. The channels cluster together in groups of 4, a tetramer.
Okay.
And each channel, each monomer, is made of 6 helices that form a sort of hourglass shape. It's narrowest in the middle.
And that's where the magic happens, the filtering.
That's where 2 key filters are. The first one is designed to stop exactly what you mentioned. Protons.
How does it do that? Because water molecules love to form chains and pass protons along, like a bucket brigade, right? A proton wire.
They do. So the channel has to break that wire. And it does this with a specific sequence of amino acids, the MPA motif.
Okay.
About halfway through the channel, this motif uses positive charges to essentially grab the passing water molecule and force it to do a 180-degree flip.
Wait, it literally turns the water molecule upside down.
That's exactly right. And that single flip breaks the continuous hydrogen bond chain. Water molecules pass through one by one, but the proton wire is broken. No protons get through.
That is genuinely incredible. The whole system hinges on just turning a molecule.
It's beautiful, but that's only one filter. There's also a physical size constraint.
The narrowest point of the hourglass.
Correct. It's called the RR selectivity filter. And this thing is tiny, only about 2.8 angstroms wide. Just big enough for a single water molecule to squeeze through.
And the RR in RR stands for arginine, which has a positive charge. What's that doing there?
That positive charge does 2 things. It helps stabilize the water molecule as it passes, but it also actively repels any other positive ion, like a stray proton that somehow got that far. It's a double-layered security system.
So you have a physical barrier and an electrostatic barrier. It's just elegant.
It really is. And this tiny, elegant structure has massive consequences for our bodies, especially in the kidney.
Right. The kidney is ground zero for aquaporins.
Absolutely. 6 of the 13 known types are found there. AQP1 does a lot of the heavy lifting for bulk water reabsorption, but the real star is aquaporin 2, or AQP2.
And that's the one that's regulated.
Exactly. It's regulated by vasopressin, or ADH. When your body's dehydrated, it releases ADH.
Which tells the kidney to put more AQP2 channels into the cell membranes in the collecting duct.
And that allows you to pull water back into the body to concentrate your urine. It's our water-saving mechanism.
So what happens when that breaks?
The results are severe. A genetic defect in AQP2 causes a condition called nephrogenic diabetes insipidus. The kidneys just can't reabsorb water. People with this can produce up to 20 liters of urine a day.
20 liters. That's life-threatening.
It is. It just shows how critical this high-speed plumbing is. And it's just as vital in plants.
For that turgor pressure we talked about, to keep them from wilting.
For turgor, for growth, for everything. But what's really fascinating in plants is how they can turn the channels off.
Gating them shut.
In response to stress, like a drought, enzymes will dephosphorylate the aquaporin proteins. That little chemical change causes the channel to physically close, stopping water from leaving the cells.
It's an emergency brake to conserve water.
A molecular drought defense system.
That is amazing. Okay, let's bring this all home. Let's tie it to the most direct, high-stakes application there is. 4V fluids in a hospital.
This is where it all comes together. Every decision about an IV bag is a decision about tonicity.
So let's break it down. First, isotonic fluids. Things like normal saline.
Right. Its concentration matches our blood, so it mostly stays in the blood vessels. It's perfect for replacing volume if someone's had a hemorrhage or is severely dehydrated. It fills the tank back up without messing with the cells.
But the risk is fluid overload, especially if the patient has a bad heart or kidneys.
Precisely. Then you have hypotonic fluids, like half normal saline. These have fewer solilutes.
So the water is going to move out of the vessel and into the cells.
Yep. This is what you use for cellular dehydration. Think of a condition like DKA, where the cells themselves are parched.
But the big red flag here is the brain.
Huge red flag. You never, ever give hypotonic fluids to someone you suspect has brain swelling. It will just pour more water into the brain cells and make the cerebral edema catastrophically worse.
Okay, and finally, the really potent ones, hypertonic fluids.
The high alert meds, 3% saline, for example, these are packed with solutes. They pull fluid out of the cells and into the blood vessels.
So you'd use this to do the opposite, to treat brain swelling by pulling water out of it.
Exactly. To reduce intracranial pressure or to treat someone with dangerously low sodium levels. But you have to monitor them like a hawk because you can easily cause massive fluid overload in the lungs.
So let's do a quick clinical scenario. A patient comes in, they're hypotensive, low blood pressure, but they're also hypernatremic. Their cells are really dehydrated. What fluid do you give first?
Biology tells you what to do. The hypotension, the low blood pressure is the immediate threat to life. You have to fix the vascular volume first.
So you start with an isotonic fluid.
You start with an isotonic fluid to get their blood pressure stable, fill the tank. Once they're stable, then you can slowly and carefully switch to a hypotonic solution to fix the cellular dehydration. You fix the big picture before you fix the small picture.
That makes perfect sense. So we've gone from basic osmosis all the way to the molecular machinery of these channels and how it dictates life or death decisions in an ICU.
It's an incredible journey seeing how this elegant piece of molecular engineering governs so much of our physiology.
I think the biggest takeaway for me is just the incredible precision of it all. We talked about how that RAR filter and the MPA motif work together just to block a single tiny proton from getting through.
Yeah.
And that microscopic decision, allowing a water molecule but blocking a proton, is directly reflected in a macroscopic outcome. It's the difference between a healthy cell and one that lizlies, or between an IV fluid that saves a life and one that causes fatal brain swelling. It's this direct line from molecular physics to medical reality. It's just something to think about.